The electron microscope is capable of magnifying up to 500,000 times. The specimen is mounted in vacuum chamber through which an electron beam is directed. The image is projected onto a photographic plate. The major disadvantage of the electron microscope is that it cannot be used to observe living objects. However, it provides a higher magnification and resolution, ability to see close points as separate, than the light microscope so that specimen can be observed in more detail. Cell structure as seen through electron microscope. The plasma membrane. Under the electron microscope, the plasma membrane is seen as a double layer. This consists of a lipid layer sandwiched between two protein layers. This arrangement is known as the unit membrane, and shows two lipid layers with proteins within. Substances are transported across the membrane by active transport and diffusion, the endoplasmic reticulum. This is a network of tubular structures extending throughout the cytoplasm of the cell. It serves as a network of pathways through which materials are transported from one part of the cell to the other. An ER encrusted with ribosomes, it is referred to as rough endoplasmic reticulum. An ER that lacks ribosomes is referred to as smooth endoplasmic reticulum. The rough endoplasmic reticulum transports proteins while the smooth endoplasmic reticulum transports lipids, the ribosomes. These are small spherical structures attached to the ER. They consist of protein and ribonucleic acid, RNA. They act as sites for the synthesis of proteins, Golgi bodies. Golgi bodies are thin, plate-like sacs arranged in stacks and distributed randomly in the cytoplasm. Their function is packaging and transportation of glycol proteins. They also produce lysosomes, mitochondria. Each mitochondrion is a rod-shaped organelle, made up of a smooth outer membrane and a folded inner membrane. The foldings of the inner membrane are called cristae. They increase the surface area for respiration. The inner compartments called the matrix. Mitochondria are the sites of cellular respiration where energy is produced. Lysosomes. These are vesicles containing hydrolytic enzymes. They are involved in the breakdown of microorganisms, foreign macromolecules, and damaged or worn-out cells and organelles. The nucleus. The nucleus is surrounded by a nuclear membrane, which is a unit membrane. The nuclear membrane has pores through which materials can move to the surrounding cytoplasm. The nucleus contains proteins and nucleic acid deoxyribonucleic acid, DNA, and RNA. The chromosomes are found in the nucleus. They are the carriers of the genetic information of the cell. The nucleolus is also located in the nucleus, but it is only visible during the non-dividing phase of the cell. The chloroplasts. These are found only in photosynthetic cells. Each chloroplast consists of an outer unit, membrane enclosing a series of interconnected membranes called lamellae. At various points along their length, the lamellae form stacks of disc-like structures called grana. The lamellae are embedded in a granular material called the stroma. The chloroplasts are sites of photosynthesis. The light reaction takes place in the lamellae, while the dark reactions take place in the stroma. Hi guys, if this is the first time you've come to my channel, please go down the bottom, hit that subscribe, and turn on all notifications so you can get notified every time I post a brand new video similar to this. Keep watching.